Sherry can take it away.
Yeah, thank you so much, Avi, for the introduction. Just to reintroduce myself one more time, my name is Sherry Shea. I'm currently a bioinformatician at the Icahn School of Medicine at Mount Sinai in New York, where it is very cold and rainy, no windstorms or heat. And today I'll be presenting on a resource we've been developing called the Dexamethasone Benchmarking resource. So to give some background on how this resource kind of came about, so we all know there's a high volume of transcriptomics data sets and related tools that are currently publicly available. And this creates a need for an objective benchmark to compare the quality of these different transcriptomics analysis tools that are used for different steps of common workflows. In essence, we want to make sure that we can maximally and accurately extract knowledge from data that's available. And in the IDG context, we especially want to be able to extract that knowledge related to understudied targets. And so this is where a silver standard benchmark built upon a well-studied genetic and pharmacological association can kind of help fill this need. So that brings us to the drug dexamethasone, which is a well-studied anti-inflammatory glucocorticoid medication. So it's structurally similar to hydrocortisone and prednisolone, which are commonly known. And this also means that there's a wealth of data related to both dexamethasone and its targets. One of those targets is the glucocorticoid receptor, GR, which is encoded by the gene NR3C1. Other targets include NR0B1 and NR1I2. And the benefit of studying dexamethasone as targets is that in addition to dexamethasone, many of its targets are also frequently studied, and there's a lot of data related to them already available as well. And even now, dexamethasone is still a relevant and widely used medication. I believe as recently as 2020, it was just approved for treatment of severe respiratory symptoms in COVID-19 patients. So to give an overview of what the dexamethasone benchmarking resource is, this is a site that contains curated datasets and Jupyter notebooks related to dexamethasone and its known targets. So these include manually and programmatically compiled datasets, and then static HTML example notebooks and downloadable Jupyter notebook files for all code that's available. And this is currently deployed on GitHub pages. The project was first started in the summer of 2022 by one of our former research trainees, Clara Chen, who really fleshed out the original concept for the project as represented here, where this resource contains compiled dexamethasone and GR datasets, as well as the code for benchmarking each of those steps in the RNA-seq analysis pipeline for each of those compiled datasets. Currently, the resource has expanded somewhat to include the following datasets. So we have dexamethasone perturbation data, including RNA-seq data from GEO, microarray data, and also L1000 chemical perturbational profiles. We also have perturbation data for dexamethasone targets. So this includes L1000 genetic perturbational profiles, as well as manually compiled ChIP-seq data from CHIA, ENCODE, and GEO. One step that we focus on benchmarking on the resource for RNA-seq data is data normalization. So this is because RNA-seq data is aligned to produce read counts, and then those read counts are usually context dependent for any given sample. Additionally, different alignment algorithms such as STAR, Callisto, Salmon, and other popular methods will produce different results. Right now, in the notebooks that are available on the site, we do benchmark CPM, log transformation, quantile normalization, and z-score normalization, as well as some combinations of those normalization methods. And we are also working on seeing if we can implement TPM and RPKM or FPKM as well. We also have notebooks that focus on benchmarking different differential gene expression analysis methods. So gene expression analysis typically will compare two experimental conditions, so disease versus normal samples, drug treatment versus placebo, genetic perturbation versus wild type. But even for the same samples, different algorithms will compute significantly differentially expressed genes slightly differently, and they each have their own system of rankings or p-value computations. Right now, the notebooks currently benchmark fold change, characteristic direction, and lemma because these are already implemented in Python. And then we're also working on including t-test as well as EdgeR and DESeq2 to add to the current notebooks. In general, each benchmarking notebook follows the same steps. So you load the data, we apply normalization if applicable, and then we apply some differential gene expression analysis method. And then in order to benchmark the results, we use enrichment analysis and generate boxplot visualizations of the enrichment analysis results. And we also look at Brownian bridge plot visualizations of the overlap between computed signatures and target genes. And I'll explain this a little more in depth in the following slides. So in general, these are the code notebooks that are currently available for download and viewing on the resource. There's 3 notebooks for benchmarking RNA-seq data for all the normalization methods, and then the signatures are computed using each of the specified methods. We have a notebook to load L1000 data, which can be run before the 2 notebooks for benchmarking, the L1000 CRISPR knockout data and the L1000 chemical perturbation data. Today's presentation will mostly show examples from the notebook for benchmarking the differential gene expression methods for the L1000 CRISPR knockout data, because we believe this data is relevant to the goals of IDG. So in general, the DEX benchmarking resource at its initial stage focused RNA-seq data. However, we realized that benchmarking the L1000 data set would be a really good opportunity. So the L1000 dataset currently consists of over 1 million chemical or drug profiles and genetic perturbational gene expression profiles. And so this includes 56 kinases, 22 ion channels, and 67 GPCRs from IDG, which are included in the L1000 CRISPR knockout perturbagens. L1000 data is also different from RNA-seq data in terms of size, quality, and content. Generally, you measure the expression of 978 genes, and then you infer the expression for an additional 11,350 genes. So the data does look different from traditional bulk RNA-seq data. And then ultimately, due to the size of the actual data set, we felt there was a large potential for drug target discovery. So we wanted to be able to develop methods to identify the best practices for computing signatures for those IDG targets that are in the L1000 CRISPR knockout data. And we also want to be able to potentially find novel targets of both clinical and preclinical drugs, as well as discover functions and mechanisms of actions for other understudied IDG genes and drugs. Additionally, we also just want to benchmark the L1000 data in general, because previously we have found that one method, the characteristic direction method, has been shown to produce good results for the original L1000 data, which was only about 350,000 signatures. But this original data set did not include any CRISPR knockout data, and we also want to see, do these results actually hold for the latest release of the 2021 L1000 data. And in this way, we can also benchmark a few dexamethasone-related signatures instead of looking at all 1 million signatures. So in general, for benchmarking the L1000 data, we really only look at differential gene expression signatures. This is because the L1000 level 3 data is already normalized. And for gene expression signature computation, as I previously mentioned, right now we've implemented characteristic direction, lemma, and full change in these notebooks. The signatures are also batch specific, so the control and perturbation samples that are used to compute each signature will have a matching time point, cell line, and experiment ID. So this would be what some of the signatures computed would look like. So you see that for each set of matching control and perturbation samples, that's represented by the columns, we can compute a signature using each of the methods that I just mentioned. So here you can see that for the same sets of control and perturbation samples, each of the signatures is a little bit different across the 3 methods. What we then want to do is we want to look at enrichment analysis against the CHIA 2022 library and enricher for each of those signatures. And to do that, we basically extract the top 250 most upregulated genes, the top 250 most downregulated genes, and then combine those two gene sets to create just the 500 most differentially expressed genes from each signature, and we submit them to Enricher. And we look at the CHIA 2022 library because this library contains gene sets created from ChIP-seq data for various transcription factors. So we're looking at enrichment of those target genes, of those transcription factors in the signature gene sets. To kind of give a little more insight into how this logically works, in general, we expect that dexamethasone perturbation or lack thereof should show a difference in the expression of its targets. Some of those targets, which are transcription factors, will in turn affect expression of further downstream target genes. So now, instead of just looking at the expression of the direct targets of dexamethasone, we can also look at the expression of the downstream targets. So then we expect that if we treat samples of dexamethasone, such as in the L1000 dexamethasone perturbation samples, or in RNA-seq studies of dexamethasone perturbation, then the dexamethasone target transcription factors generally will be activated. Thus, any genes that are downstream targets of those dexamethasone targets themselves should also exhibit some change in expression. So in this case, we can see that NR3C1 is a transcriptional activator. So we expect that when it is activated, its target genes in turn should also be upregulated. And on the right, you can see the enricher results when we submit the up genes and the down genes for dexamethasone perturbation, that NR3C1 is ranked higher when the up genes are submitted, indicating that NR3C1 target genes are enriched in the upregulated genes that are under dexamethasone perturbation compared to the downregulated genes. On the flip side, if we knock out any of the dexamethasone targets, and this is where the L1000 CRISPR knockout data would come in, we would expect that the target genes or those transcription factors would again display some sort of differential expression. In this case, I have example enricher results for NR1I2. So it is a transcriptional activator, and we expect that if NR1I2 is knocked out, then those target genes of NR1I2 would generally be downregulated in those knockout samples. And we can see that the downregulated genes, some of the NR1I2 knockout signatures, do seem to rank NR1I2 higher than the upregulated genes. So then what this leads to is that we can take the average rank of those knockout gene terms for each of the knockout gene signatures and then create box plots, kind of visualizing where each term ranks for each of the different gene sets from each signature, computed using the different methods. So here I have the example box plots for NR1I2 knockout perturbations. So on the left, the box plots are just colored by the direction of the gene set, and on the right, they're colored by the method. And you can see that NR1I2 is an activator, as I just mentioned. So you see that the down-regulated gene sets all tend to be very enriched for NR1I2 target genes, whereas the up gene sets are very not enriched for the NR1I2 target genes. And it's important to note that there is a signal here that's being picked up. These results are not random. There is a random box plot that's just taking random gene sets and submitting them for enrichment analysis and seeing where NR1I2 ranks. This method also helps us identify differences in the functions of different transcription factors, because here we see that for NR0B1, the results look slightly different from NR1I2 knockout. So NR0B1, most evidence shows that it tends to be a negative regulator. So here we actually see that the upregulated gene sets in the NR0B1 knockout signatures are actually highly enriched for the NR0B1 target genes, whereas the downregulated gene sets are not. And once again, we do see this pretty consistent signal, and it's very different from what we'd expect if these signatures are just randomly ranking the genes. Finally, the last step in the notebooks, as I mentioned early on, there are these Brownian bridge plots or random walk plots that are used to compare each signature to the target gene sets, for a knocked out gene. So essentially at each step, we want to rank the genes in each signature from highest lowest expression value. And then at each rank, we want to check whether the gene is actually in the target gene set for the knocked out transcription factor. And so what we expect to see is that if the genes in the target gene set are being recovered, then we should see this nice positive arc in the graph. Here we can also see that the characteristic direction method has all of the methods are performing pretty well because we see that nice arc, but the characteristic direction method does seem to have a slightly sharper incline at the very early ranks. So then based on the benchmarking results, these are just preliminary results based on the notebooks we have now. We see that the characteristic direction method does seem to be performing pretty well for differential gene expression analysis of L1000 CRISPR knockout data. And so that leads us to our case study, in which we can look at an IDG target, which in this case is the understudied GPCR GPR183. So here's a screenshot from a new resource our lab is developing called Gene Ranger, which shows that GPR183 is highly expressed in immune cells across transcriptomics data from ARCHES-4 datasets. To dig deeper into the mechanism of GPR183, we can then compute a signature from the L1000 data for the CRISPR knockout of GPR183 using the characteristic direction method. And then we can query the signature to identify other CRISPR knockout signatures that may have a similar signature to GPR183 knockout. So this graph shows the results of querying the signature in SIGCOMM links with the up and down regulated genes from the GPR183 knockout signature in the A375 cell line. We can then take the top member gene knockouts to construct a cell signaling pathway that hopefully might shed some light on the molecular mechanisms of GPR183. So essentially, we can take the top 20 most similar knockout genes, GPR183, and then we can submit these genes to Enricher and visualize the enrichment results as a knowledge graph using the Enricher KG platform, another new app that our lab is developing. Here, the green nodes indicate the top knockout genes of signatures that are similar to GPR183 knockout, which were the actual input gene set. And each of the other nodes denote terms from each library based on the legend that are enriched for these genes. One interesting finding we saw is that TGF-beta2 is one of the genes with a similar knockout signature to GPR183, which suggests its potential involvement in GPR183 signaling. Additionally, we also found 4 kinases, DGKQ, HIPK3, CAMK1G, and DIRK2, which are present in the top 20 most similar knockout signatures to GPR183, which suggests potentially a novel cell signaling pathway downstream of GPR183 that could be involving these specific kinases. So in general, the Dexamethasone benchmarking resource is currently being developed to provide a silver standard of datasets and code for comparing the quality of transcriptomics analysis tools. And this is just a first pass at identifying the best practices for analyzing transcriptomics data in general. So we hope to continue to improve and build on this resource. Providing benchmarks for RNA-seq data, as well as the L1000 data, expands the potential for new knowledge discovery from data that's already existing and publicly available. And the addition of visualizations and the use of interactive code notebooks makes this pipeline easily accessible and reusable. The methods outlined in the resource can also help guide the investigation of understudied IDG targets, such as GPR183. And eventually, we hope to use the resource to also examine how L1000 chemical perturbation signatures combined with the CRISPR knockout signatures can actually be used to identify small molecules that may directly or indirectly target IDG targets. So with that, I'd like to thank all former and current members of the Mayan Lab, as well as all of our funding partners, and thank you for your attention today.